Okay, get this. What if the secret to the future of AI is already written inside your own DNA? In this explainer, we're diving into a really mind-bending connection between the code of life and the code that powers artificial intelligence.
And yeah, I know, that sounds a little sci-fi, right? But the answer is surprisingly deep. It points to this universal principle of how intelligence works, whether it's in us or in a machine. You see, it's not just about having the code. The real magic is in how you use that code.
All right, so to really get this, we've got to start with a problem. A huge problem, actually. One that both Mother Nature and AI engineers have been wrestling with forever. And the problem is this. How do you build a system that's stable, reliable, but can also adapt to a world that's constantly changing?
Think about it. You got this massive, fixed blueprint. For us, it's our DNA. For an AI, it's its giant base model. That's the stable part. But then, bam, the world throws a curveball. So what do? You can't just rewrite the whole blueprint from scratch every single time. That's super slow, incredibly risky, and chances are you just end up breaking the whole thing.
Well, biology's solution is just beautiful. It's so elegant. Instead of rewriting the hardware, it built a dynamic operating system to run on top of it. And we call this layer the epigenome.
So what is the epigenome? Basically, it's a bunch of chemical marks or tags that get stuck onto our DNA. The best way to think about it is like sticky notes on a giant textbook, right? The tags don't change the words in the book. The DNA sequence stays the same, but they tell the cell which pages to read, which ones to ignore, and which sentences to really pay attention to. It's all about controlling access.
And here's how that actually plays out. Most of the time, your DNA is wound up super tight, like a spool of thread. This keeps genes silent, turned off. But then, a signal comes in from the outside world. Could be from food, stress, anything really. That signal causes a chemical tag to latch on. And that little tag makes the DNA spool loosen up right at that spot, exposing a gene. Suddenly, the cell's machinery can get in there and read it. It's this incredible, dynamic switch that lets us adapt in real time without ever having to mess with the core DNA code itself.
Okay, now here's where it gets really wild. AI developers who were up against the very same stability versus adaptability problem, totally independently, stumbled upon almost the exact same solution. This is a textbook case of what biologists call convergent evolution, two different paths leading to the same brilliant answer.
For AI, this solution is called the context engine. You know, when you type a prompt into an AI, you're not actually reprogramming it, not at all. What you're doing is giving it a temporary set of instructions, a context. You're telling it, hey, out of all the billions of things you know, focus on this part of your brain for a minute. Your prompt is literally acting like one of those epigenetic tags. It's guiding access without rewriting the library.
And the source material for this explainer has this just perfect quote. It says, "A well-crafted prompt is acetylation writ large." Now, acetylation is just the fancy biological term for adding one of those chemical tags. So the prompt loosens the chromatin, the coiled-up data, of the AI's latent space, which is basically its huge internal map of concepts. What's that really mean? It means your prompt makes the right information easy to grab exactly when it's needed. So let's actually put these two systems side by side. Because when you do, you see this isn't just a cute analogy. The connection is, well, it's shockingly deep. The different parts map onto each other almost one-to-one.
Okay, this table, it's basically a Rosetta Stone for understanding intelligence. Check it out. The fixed DNA sequence, that's the AI's base model weights. The epigenome, that dynamic control layer, that's the AI's attention and context system. Those biological signals that kick things off, the transcription factors, that's your prompt, or data from a system like RAG. And even the tools that do the modifying. In our cells, they're called chromatin remodelers. In AI, they're things like LoRa or adapters that fine-tune performance. I mean, come on, the parallels are just staggering.
So what's the big idea here? It's this. Real intelligence, the powerful kind, isn't just about how much information you can store. It's not about the size of your library. It's about being able to control the gradients of accessibility. Fancy words for dynamically tuning how easy or hard it is to get to the right piece of info at exactly the right time.
And get this, the analogy holds up even when things go haywire. Because what happens when these super delicate regulatory systems fail? Well, in both biology and AI, a breakdown in regulation is catastrophic.
Think about cancer. At its core, it's often a failure of epigenetic off switches. Genes that should be silent are turned on, and cells grow out of control. Now look at AI.
It has a similar problem called mode collapse or reward hacking. The AI finds one answer it thinks is good and gets stuck on it, repeating it endlessly in a hallucination loop. It's an uncontrolled activation of one part of its system, just like cancer.
Or how about aging? As we get older, our epigenetic patterns can get a bit messy. The regulation isn't as crisp as it used to be. Well, AI models have the same issue. It's called model drift or catastrophic forgetting. Over time, their performance degrades, their answers get less sharp, and they can lose skills they once had. In both systems, the regulatory layer is just slowly wearing out.
So, what are we supposed to take away from all this? The point is, this isn't just some clever party trick or a neat analogy. It's evidence. It's pointing towards a universal principle of how complex, adaptive systems have to work. It's that idea of convergent evolution again. Life and AI, two totally separate domains, independently discovered the same fundamental solution to the same fundamental problem.
But why this solution? Why not just rewrite the code? Well, because trying to edit the core blueprint, whether it's DNA or the base model weights, is just a terrible idea. It's incredibly high risk, most changes will break something, it's painfully slow, and you might accidentally erase decades or millions of years of hard-won knowledge. Having this flexible, reversible layer on top, it's just smarter. It's a more efficient, more elegant way to adapt.
And that brings us to what might be the single most important idea in all of this. It's summed up perfectly in this quote: "Intelligence isn't omniscience, it's masterful, selective access." It's not about being a know-it-all. It's about being a master of focus, knowing what to pull up and what to push down at exactly the right moment. So here's the big takeaway for all of us. For the longest time, the story of AI has been about one thing: scale. Bigger models, more data, bigger libraries. But this whole parallel with biology suggests that might be changing. Maybe the future isn't about building a bigger library. Maybe it's about training a much, much smarter librarian. Which leaves us with a pretty provocative question to chew on. Are we witnessing the end of the brute force era of AI and the beginning of a new one focused on elegant, dynamic regulation? Because this shift from just having static knowledge to having adaptive, masterful access, it's not just a trend in AI. It's forcing us to rethink what intelligence is, period, both in the machines we're building and in the biology that built us.